Okay, now let's take a look at amino acids and their basic properties and how we can combine amino acids chemically into making proteins. So we'll entitle this Amino Acids and Proteins. To begin, let's take a look at the basic structure of an amino acid. There are, of course, 20 amino acids that are used to make proteins. Recall from your molecular biology class that this is adds to the diversity of proteins because the sequence is based on the gene for the individual protein, and the sequence of amino acids is the characteristic of the protein itself. Now, of these 20 amino acids, each one has a different side chain, which is also called an R group. This is the side chain, and this is what gives the unique properties, unique qualities and characteristics to each amino acid. Now, the basic structure of an amino acid is we have a primary amine connected to a carbon, which is considered the alpha carbon. This carbon has a hydrogen bound to it, and it also has a carboxyl group bound to it. And finally, the fourth bond of this carbon is the R chain, which is designating to the particular amino acid. Now, notice that there are 4 different groups bonded to this central alpha carbon. That means that this carbon is a chiral center. This is true for 19 amino acids, because one amino acid, the R group, equals a hydrogen, which is called glycine, has the same as the hydrogen bound to the alpha carbon also, so that is not a chiral center. Now, being a chiral center indicates that there is a handedness in the stereochemistry of an amino acid. So amino acids can either be in an L, levorotary, or a D, dextrorotatory configuration. Now, there are 2 ways to draw this configuration. The first is in the Fisher projection, which is what I have here. And the way to draw this, we will place this carbon here, and then we will have our carboxyl group, our alpha carbon. We will have our R group come off the bottom, and the hydrogen to fill out the 4 bonds on the alpha carbon coming off to the right. Now, when the amino acid is drawn in this structure with the R group in a linear form with the alpha carbon and carboxylic acid, carboxyl group, the placement of the nitrogen, the primary amine, will determine the configuration of the amino acid. So, for example, if the primary amine connected to the alpha carbon is on the left in this Fisher projection, this is the L configuration. If, for example, we draw our amino acid in a slightly different form, where we have the primary amine on the right-hand side of this Fisher projection, this would be the D configuration. Now, another way to look at this is what I like to call walking the bridge from the carboxyl group to the amine. So what does that mean? If we have the central alpha carbon drawn in this configuration, and these 2 bonds are in the plane of this paper, we can draw our nitrogen here and our carboxyl group here. Now for the other two bonds, the stereochemistry, the 3D chemistry here, will be determined by the L or D configuration. If we are in the L configuration, that would mean that we have the R group on the left-hand side. So I'm going to draw this with this triangular bond structure and shade this in, indicating that this is coming out of the paper, coming toward us. If this is the case, the fourth bond on the alpha carbon, being the hydrogen, is going to be going in to the paper. Now this configuration, when I say walking the bridge from the carboxyl to amine group, is if we were to see this as a bridge structure and we start walking in this direction, whatever our hand would touch walking across the bridge, whether the R be on the left or the right, will be the configuration. So this drawing here of this amino acid stereochemistry shows that we would walk over this bridge from the carboxyl to the amine. Our left hand would touch the R group. This is considered the L configuration. If the R group, in this case, and the hydrogen were flipped, then the R group would be on the right-hand side, and walking the bridge from the carboxyl to the amine, our right hand would touch the R group, which would be the D configuration. Now, why is this important to understand? And that's because all of our proteins are constructed of L amino acids. This is an important point when we consider protein structure, when the protein begins to fold after the amino acids are linked to form the final protein. The L configuration will place the R group in a certain position that it will need to eliminate steric hindrance or steric effects from the adjacent amino acid R group. And based on the R group being placed in the L configuration, this is going to generate a right-hand turn. To the protein. We'll go over this more when we talk about protein structure. Now, there are 20 different amino acids, and it's very important to take a close look at each one of the side chains. And these side chains are broken up into multiple functional groups. They can be hydrophobic amino acids, have hydrocarbon R groups. And recall, hydrocarbon R groups can form hydrophobic interactions and bonding. So hence, these amino acids with these hydrocarbon side chains are considered hydrophobic amino acids. We have other amino acids that will fall into the category as polar. Polar here means that there are strong electron withdrawing groups that can form partial charges and partial charge distribution. These tend to have hydroxyl groups. Or amides. Notice that this can form a partial charge, giving its polar characteristic. We also have sulfur containing amino acids. These are the two methionine and cysteine. And we also have charged amino acids. These charged amino acids can either have carboxyl groups at the end of the R chain, or they can be basic, having an amine at the end of the R chain. Now, what's important to keep in mind also with amino acids is if we look at the structure here of the original backbone of the amino acid, is that we have the potential for charge on an amino acid. So charge means that we can titrate these functional groups using acids and bases, and we can change their chemical properties such that we can protonate or deprotonate depending on the pH. The R chain, if it has a charged group to it, or it has an ionizable group, such as a polar or a sulfur-containing group, these also can be modified based on the pH. So let's take a look at the titration curve of a particular amino acid. And let's start out with a simple one. Alanine. Now alanine, if we have no hydroxide ion present, alanine will be in its most acidic form. And its most acidic form is going to be in the fully protonated form. The R chain, of course, with alanine is going to be a methyl group. So for alanine, we are going to be titrating two functional groups because the side chain does not have a charge and it doesn't contribute to the titration properties. So in its pure acid form with very little or no hydroxide ion, we have a fully protonated form of alanine. The charge on this particular molecule is +1 because the amine is protonated and so is the carboxylic acid. Now as we add hydroxide ion, we are going to raise the pH of the solution and we are going to reach an inflection point. As we saw with our titration previously of acetic acid, the inflection point is going to be equivalent to the pKa. In this case, this is going to be considered pKa 1. If we continue with our hydroxide ion titration, we will see that our pH starts to climb after we are past the buffering capacity of this particular group, and then we will reach another inflection point, which will be pKa 2 for our second titratable group. And again, continuing with our hydroxide ion, the pH will dramatically increase after we move through the buffering capacity of this particular functioning group. Now the question is here is which pKa corresponds to which functional group? Well, think of this as which proton is the best leaving group. The best leaving group, the best leaving ion, will be the one that is bound the weakest. Now consider this hydrogen bound to this nitrogen. This electronegative atom will be pulling electrons to maintain the binding here of the proton, but this is a fairly stable bond. Now look at this bond here. Notice we have in the carboxyl group, we have the potential for resonance between these two oxygens on this carbon. These two sharing electrons is going to be electron withdrawing from the oxygen-hydrogen bond. That means this bond between this oxygen and hydrogen will be very weak. If it's very weak, it will not take very much hydroxide ion to extract this hydrogen to form water and leave this particular ionizable group in its negatively charged form. So the pKa, the first pKa here, is 2.34. And the definition of this pKa1 is that at a pH of 2.34, 1/2 of these carboxyl groups will be in its protonated form, 1/2 of these will be in its deprotonated form. So at this point, we have a charge of -0.5 contributed by the half deprotonation of this carboxyl group, and +1 contributed by this amine, which will still remain protonated largely at this pH. So this will give us a charge of 0.5, positive 0.5 on the amino acid. Now if we move up to the pKa2, pKa2 is the pH at which the next ionizable group will be represented by 50% in its protonated form and 50% in its deprotonated form. Since we've already taken care of this ionizable group, we're going to be looking at this amine. This pKa is 9.69. And at this point, what would be the charge on this carboxylic acid at this pH? This is such a basic pH, and this is such a weak bond, we can pretty much assume that the carboxylic acid on the alanine amino acid is going to be fully deprotonated. So this will contribute a charge of -1. Now, the definition of pKa2 for this ionizable group means that half will be protonated, half will be deprotonated. So that means at this pH, 9.69, this group here will contribute positive 0.5 charge, because half of these ionizable groups will be charged and half will not. So now we have a total charge on the amino acid of -0.5. Now an important consideration, again, and as we have seen with our acid and base titrations in a previous video, is determining the pI, which is the isoelectric point. And this, of course, is the point where the charge on the species we're looking at, in this case is alanine, the charge is equal to 0. So at pKa1, when we achieve a pH of 2.34, we will have an overall charge of positive 0.5. At pH 9.69, we will have an overall charge of -0.5. So the question becomes then, at what point, at what pH along this range is alanine representing a 0 charge, balanced positive with negative? That is going to be halfway between these two. So the pI equals the pKa1 plus pKa2 divided by 2. And at this value, at this pH, we have an equal balance of the positive and negative charge, which will lead an overall charge on the molecule to be 0. Now the other information on this video we need to examine is peptide bond formation. In order to make a protein, we need to couple the amino acids together to form a linear chain. So amino acid 1 will need to be coupled to amino acid 2, amino acid 3, amino acid 4, etc. Linking these amino acids together will form our protein. And of course, the R chains off of each one of these amino acids will be characteristic for the protein. And this particular sequence gives the definition of what the protein is. The sequence of amino acids that form a protein equals what is considered the primary structure of a protein. So the primary structure is essentially the sequence of amino acids that define what the protein is. Now, how do we link proteins together? The key here is understanding some basic chemistry. I will draw the amino acids, 2 of them together, in their respective configuration, ionic configuration, that we would expect at pH 7. Now recall, pH 7 is our biological pH where most of our proteins will spend their time and have their highest level of activity. So at pH 7, refer back to our titration curve, pH 7 is going to be approximately right here. So if we look at this configuration, ionic configuration of our amino acid of alanine at pH 7, notice how close it is to the isoelectric point. This essentially means that the amino acid will have a 0 charge because these 2 ionized groups will balance each other out. Now we are going to take a second amino acid, this would be considered amino acid 2, and we are going to form this bond between amino acid 1 and amino acid 2. This is very simply a dehydration reaction. The nitrogen here is going to be a nucleophile, and it is going to bind to this carbon, and we will form our coupled amino acids. Here is the new bond we have formed. The displacement of this oxygen will be through 2 of these hydrogens. 1 hydrogen remains, giving us our stable 3 bonds on nitrogen. These 2 hydrogens form with this oxygen, making water, H2O. So this is a simple dehydration reaction to form this bond, which is called the peptide bond. We'll talk more about the interesting characteristics of the peptide bond when we talk about protein folding. Another note to note about this dehydration reaction is that this is not typically a favorable reaction. And so therefore we need enzymes to overcome the free energy of activation of this reaction, and these enzymes are housed, of course, in ribosomes. There's also energy that goes into the ribosomal activity, and this is using an energy storing molecule called GTP. We'll look at this more later in a later video. Now, in order to lengthen this structure, we will just add another amino acid onto the what's considered the C-terminal end for carboxyl terminal end. Eventually, we will make the full length of the protein as designated by the gene of the protein. Now, amino acids and proteins come together, and these proteins can form very small or very large structures. For example, we'll talk about one particular protein in energy production called cytochrome C. This has 104 amino acids. This is considered a very small protein. Other proteins, such as titin, which is the largest protein identified in humans, has 26,926 amino acids.